In this session, we are going to study about the seed, the structure of a seed, and the types of seed found in monocots and dicots. So let us start with the seed. Now, if we talk about the seed, we have studied earlier that the ovules after fertilization turn into the seeds, and the ovaries, they turn into the fruit, right? So the seeds are formed from the ovules after fertilization. Now, if we talk about the structure of a seed, then a seed usually have two parts: a seed coat for its protection and an embryo inside it, which generally give rise to a new plant. So a seed basically has two parts: seed coat and an embryo. And if we talk about the embryo, it has several parts like plumule, which gives rise to the shoot, and an embryonal axis, then it also has a radicle which give rise to roots. We studied about this in the previous lecture when we studied about the stem and the root. We learned that they are formed from the plumule and radicle respectively, right? So they are the part of the embryo. And besides that, we will also find cotyledons in an embryo, and these depending on the type of the plant, the cotyledons or the seeds may be of two types: monocots and dicots. In monocots, only one cotyledon will be there, in the embryo, while dicots will have two cotyledons. So we will study about the monocotyledonous and dicotyledonous seed along with their structure and examples. Alright, so let us start with the structure of a dicot seed. Now, if we talk about the dicot seed, then there is an outermost structure in the seed which is called the seed coat. Now let me draw one seed so that it's easier to understand. So suppose this is a seed, a dicot seed, then this outer covering is called the seed coat, which provides protection to the seed. And this seed coat has further two parts. The outer part is called the testa, and the inner part, which is called the tegmen. Alright, so testa is the outer part and tegmen is the inner part. Both of these constitute the seed coat. Then on the seed coat there is a scar like this. This scar is called hilum, which represents the place at which the seed was attached to the fruit. Alright, so when the seed was developing, it was attached to the fruit at the place or the spot which is called the hilum. Now above the hilum, we will find a small opening or a pore which is called a micropyle. Through this micropyle, the water enters inside the seed and helps the seed to germinate. Alright, so that is the function of micropyle. I hope seed coat, hilum and micropyle is clear to you. Now within the seed coat we will find an embryo, and let us study about the structure of a dicot embryo and its various parts. So a dicot embryo has an embryonal axis This is the embryonal axis. Alright, and since it is a dicot seed, it has two cotyledons. These are the two cotyledons. Alright, so let me write it here. This is the embryonal axis. These are the cotyledons. Now if you remember, the lower part of the embryo from which roots develop, that's called a radicle. And the upper part from which the shoot develops is called a plumule. Right? So this is the typical structure of a dicot seed or a dicot embryo. Now if we look at the cotyledons of this embryo, it is quite fleshy and big. So why is it fleshy? Because it stores food material. The cotyledon stores food material. That's why when we eat the dicot seeds, we get nourishment or the nutrients from this stored food material. Now what is this stored food material actually made up of? It is actually consumed by the seed or by the embryo while growing. There's a special structure called endosperm which develops after fertilization. There's a special type of fertilization called double fertilization which results into the formation of endosperm. And that endosperm nourishes the developing embryo inside the seed. So when that embryo consumes the endosperm, that consumed endosperm is converted into these cotyledons. The stored food material comes from that endosperm. So I hope it is clear to you. Right? Now sometimes okay, so what happens when these cotyledons are fully formed in dicot seeds? No endosperm remains inside the seed because all the endosperm is consumed by the developing embryo. So such seeds are called non-endospermous seeds. Non-endospermous or non-albuminous seeds. Those seeds which do not have endosperm because it is completely consumed by these cotyledons. So it is usually the dicot seeds are non-endospermous. So the examples of dicots are pea, bean and gram, etc. So if you open out a pea, seed of a pea or bean or a gram, you will see this kind of structure. It will bear two cotyledons and there will be a small embryo at the middle, and you can easily see how big these cotyledons are, and there will be no endosperm. Hence they are called non-endospermous or non-albuminous seed. Alright, so let us now move on to the next type of seed, which is the monocot seed. So let us learn about the structure of a monocot seed, monocot embryo to be precise. Now in monocots, the endosperm is not entirely consumed by the developing embryo, rather it sustains. So monocot seeds are endospermic. Or albuminous. But there are some exceptions like orchids. So orchid seeds are non-endospermic. Alright, so let us draw the structure of monocot seed or embryo. What happens in this is that if we talk about the monocot seed, the seed coat is very membranous and fused with the fruit wall. The seed coat is membranous and fused with the fruit wall. So we can say that the fruit is not very fleshy, or the fruit parts of the fruit wall, there's nothing there, just the seed coat and the embryo present inside it. And then there will be endosperm, right? Now, if we look at the structure of the embryo, there will only be one cotyledon because it's a monocot seed. So I will draw it a bit in a slant way. So if this is the embryo, then it will only have one cotyledon which is not well developed. Monocot seeds have only one cotyledon which will not be very well developed, right? And endosperm will be present because the endosperm is not entirely consumed by the developing embryo. So above this cotyledon we will have a layer of endosperm which is not consumed by the developing embryo. So let me label it. This one is your cotyledon. This cotyledon has a name. It is called a scutellum. Then this will be the endosperm. Now the endosperm has a layer, a protective layer, which separates it from the embryo. The embryo forms a very little part at the corner of the monocot seed. The majority of the part is taken by the endosperm. And this endosperm has a protective layer or covering over it, which is proteinaceous in nature. It is made up of protein. And this layer is called aleurone layer. Aleurone layer. Okay, now this is the embryonal axis. Now, just like the dicot seed, the upper part of this embryonal axis will be called a plumule, which will form the shoot, and the lower part will be called a radicle. Now, in case of monocot seeds, some additional coverings are present in the form of sheath, which covers the plumule and the radicle. So, near the radicle, a sheath will be present, or a covering, which is called coleorhiza. And above the plumule, similarly, sheath will be present, which is called coleoptile. Alright, so this is the structure of a monocot seed, and within it the monocot embryo. We have learned that the monocots have endosperm because the monocot embryo, the embryo inside the monocot seed, do not consume the endosperm completely. Therefore, the only one cotyledon develops, which is not very well developed, and that is called scutellum. Alright, and the endosperm remains, which forms the majority of the monocot seed, and the embryo is there present at the corner, does not take much of the space, or with only one cotyledon. The endosperm has an outer covering made of protein, which is called aleurone layer. Then the plumule and the radicle of the embryo have additional coverings over them, which are called coleoptile and coleorhiza respectively. Right, now there is some exception in monocot seed which do not have endosperm, and those are called, the example is orchid. Usually monocot seeds are endospermic, but orchids are non-endospermic. Alright, so I hope this is clear. Some other examples of monocot seeds are maize, rice, and castor. Alright, so this completes the session about the seed and the types of seed, along with the examples and the diagrams and their structures. I hope all the terms is clear to you. That's all for this lecture. Thank you so much.